Okay, so chapter 8, I will try to get the quiz done today and up, but again, chapter 8, quiz is not till you get back, exam not due till you get back. I'll make everything due on that 21st, unless it's the presentation for the grad students. That was changed, that's now a week later, so the 28th of March. Okay, also, I'm not entirely sure why I put chapters 10, 11, and 12 on as part of the syllabus, because I've never presented it before. So we will either stick with those or we will be moving into the Hallmark papers, because there's 3 of them, and having a month really to do 3 papers, those dense, isn't a lot of time. So I'm going to figure out how I'm going to handle doing chapters that I haven't presented on yet and handling the papers. Okay, so we're going to get into chapter 8. There we go. So with cancer, there can be spontaneous remission. Nobody understands why. That's all I can say about that. Happens. All right, but when it comes to understanding diagnosis, things like that, the one thing that's helpful, and everyone I think wrote about this, like biopsies are one of the first things to do when you suspect a cancer is there. So we already know that looking for a large nucleus, right? That's one thing. You're also looking for change in morphology. That's how the cell looks. So it's now changing from what its regular shape is and maybe going back into a stem cell type shape before differentiated. So what we're seeing in A is this is a DNA stain. So the dark purple dots are the staining of the DNA. And you see these normal fat cells. Everyone has had that on like an A&P quiz or exam before. Fat cells should just be these giant globules of whites, or depending on your stain, black or blue, but they should be big and not. That's the only thing you can see. But when we hit this cancerous growth, you can see tons of DNA. So we know that there's extra copies of DNA. If you were going to be someone who looks at biopsies all the time, then you know exactly what you're looking for. For us, if we didn't know that there was a stain for DNA, we might think that's totally fine, that it's fat cells in reticular tissue. So again, it's basically a trained eye in order to tell if something's cancerous. On B, what's great about B is it's a stain for a hormone receptor. But this is breast tissue. Oh, I can't sell breast. Okay, whatever. I think I wrote beast tissue. Breast tissue. And when someone is diagnosed with breast cancer, they always talk about, is it triple negative? Well, one of the triple negatives they're talking about is the estrogen receptor present. We'll see that that's very helpful if the estrogen receptor is present in terms of treatment. So if it's not present, then it's considered negative. And so from this slide, we can say that this individual is ER negative. So we can't say the triple negative, but we can say 1 negative, because this does not stain for the other types of proteins that we're looking for. Okay, so that is helpful as far as treatment that is available. If this was ER negative, well, we couldn't use a chemical that blocks the estrogen receptor or binds to the estrogen receptor. But since it's ER positive, this is really good. So you've got the receptor that can bind a chemical that maybe stops proliferation. So this is helpful to courses of treatment. For C, we're looking at an antibody for epidermal growth factor. So EGF. That's a hormone, but remember, some hormones are proteins. And they're just looking to see, in this area, is there a lot of epidermal growth factor hormone? If there is, it means that these cells are being told to go through cell division because there's so much growth factor in the area. So when you see a stain like this show up, then you go, well, here those cells are being told to divide, but through this area, we're not seeing epidermal growth factor, so those cells are not. Again, helpful to, in terms of treatment, if they did this stain and then also did an epidermal growth factor receptor stain, then they could see if the receptor and the epidermal growth factor were happening in the exact same place. If that's happening, then you know for sure those cancer cells have the receptor for epidermal growth factor and will start to divide a lot. Okay, so that's another way to see what's happening to that tissue and what type of treatments would be best. And then D goes back to looking at DNA. But in this particular figure, they were staining for a very specific gene. So they're not staining just for all DNA. Let's just see if the nucleus is bigger and there's lots of more DNA. They're probing for a specific gene. And if they see in a single cell multiple copies of that gene, then we can assume that there's been some dysregulation in maybe that mitotic spindle, and some cells have more chromosomes. So we're looking at extra chromosomes maybe, but certainly extra copies of genes. And if you make it a probe towards an oncogene, and you see that you have extra copies of that oncogene, then you know that's really bad, that those cells are really trying to push forward with division. It's hard, and I can't really zone in, but this cell right here, you can see 2 purple dots in there. Some of the others, you can see 3. But again, you really just you don't want to see many purple dots. Okay, so that will help diagnosing what type of cancer you have and then possible treatments. Obviously, looking at the size of the tumor is going to be helpful in determining what stage you're in. So I think somebody gave a presentation on this, one of the grad students, going more in depth on, well, how big is the tumor? Has it moved to lymph nodes? And then has it even moved farther off throughout the body? All right, so why point out this graph? This graph is showing that it takes a long time for us to be able to palpably feel the cancer, the tumor. So this whole time, that tumor's been growing and growing and growing, and it's not till here that we can actually feel it. But then look how short of time it is between feeling it and dying. That's a much shorter time. So we know that it takes time for cancer to form because we have to go through many years of chromosome duplication and then also dysregulation. We talked about heterozygote genes and the phenotype coming out from those recessive genes. So you can see a lot of different things have to happen over time. Finally, it's big enough, and then you die. No, none of us die because we know about cancer and we're going to get proper treatments and checkups. Okay, so different types of surgery that can occur. If it is quite small, the tumor is quite small, and you can really see where the tissue has turned into cancers, then it is easy to take out a small tumor. So extirpate is just the fancy word for the removal of all the cancer cells. Lumpectomy would just be removing some of that tissue and a little bit of the surrounding area. There's a reason why you need to get a little bit of that surrounding area. And this is just showing, this is very common for DCIS, ductal carcinoma in situ. Okay, so easy to remove. Okay, why we talk about removing the tumor and some of the nearby tissue. So here's a tumor. I'm just going to draw a bunch of cells. We got a tumor going by. And then here's all the stroma. So this is just the tissue that the tumor is in. All that blue area is considered the microenvironment. These are the normal cells that are growing around the tumor, but the tumor is actually usually directing what's really happening in that microenvironment. It might be releasing growth factors to grow the tumor. It might be releasing factors to increase angiogenesis to the cell, to the tumor. Microenvironments also can be hostile to white blood cells, right? White blood cells, natural killer cells, want to go kill that tumor, but if the microenvironment is hostile to them, then it won't allow it. They're usually have a pH that is slightly different. Their oxygen levels are different. So it is a small little environment that is not normal for your cells to be around. So you've got this whole microenvironment, you've got this big tumor. What can happen is that you'll have small little satellite tumors around the area. So when you do some type of lumpectomy, while they say they don't want to remove, cause more damage, but they still need to get a little bit of that microenvironment to make sure those small tumors are gone. So here's a issue with a tumor that's really big. A really big tumor has implanted itself into the body. You've got a lot of extracellular matrix. So this whole area around here is its microenvironment. And what can happen here is this big tumor obviously has made this blood cell go into it. So you run into issues when you do larger removal of a tumor just because of the vascularization that's around that area, and then it's healing. So one way of treating this would be to really kill off the actively growing cells, which are on the outside of the tumor. The really active cancerous cells are on the outside. Inside, which is interesting, inside this tumor, these cells are dormant. They're no longer growing. They are silent. They're not dividing. They're not even giving signals to divide. So if you are to remove more of the outer cells, then you're decreasing what's known as the burden of the tumor on the body, because the inside cells are dead and they're not going to call in all those vascularization and all those other epidermal growth factors. So try to remove the giant portions on the outside, and the dead cells will probably just stay dead. Probably. So prophylactic surgery, or anything prophylactic, is trying to prevent something from happening. So I think everyone's aware of the Angelina Jolie type situation where some women who have breast cancer in their families, so it's very likely that they'll get breast cancer, or they've even had the genetic testing done and they know for a fact that the BRCA1, BRCA2 genes are mutated, then they may choose to have full breast removal of either both or one of the breasts. So that's like a prophylactic surgery to try to prevent the disease from even occurring. So mastectomy is one prophylactic. Another thing is prophylactic hysterectomy, removal of the ovaries, because that is also with BRCA1 or BRCA2 mutation, high areas of cancer incident. There are other types of prophylactic surgeries, but those are like the most common ones. Okay, so this again is kind of showing you that big tumors will call in blood supply to it. So what we can do, so what this tumor is going to do, you see these tiny microtumors right here and here, and that's essentially what I was drawing in the other figure. Where did I draw it? Right there. So the big red thing, and then you have those smaller tumors. That big tumor doesn't like to play with other tumors. So it will, in fact, it has its own blood supply. It's going in. Those cells are going to release an endostatin, and that inhibits angiogenesis. So it's trying to starve that tiny microsatellite grouping of cells. That's its hope, is that it starves that out and it continues to live off the blood vessels that's already in there. So if we were to remove this giant tumor, then these little microtumors, if we didn't catch those, then they no longer have that endostatin being released by the giant tumors, so there's nothing to stop these little microsatellite tumors from growing. Really important when you remove a big tumor or a tumor from that area, that you monitor and even do continual staining. Sometimes they'll do biopsies or removal of a tumor slice by slice, and they keep checking the stain. Okay, is there still a high epidermal growth factor level? Or, if as you go down the slices, once you no longer see the epidermal growth factor or some type of angiogenesis inhibitor, then you can say, oh, I have fully removed that tumor. Now, as I slice a little bit below, are there more tumors in that area? So it's always an area you got to continually check several months to see if something new has started to grow. All right, moving into different types of treatment. We know that X-rays are a way to induce cancer, right? We can do tons of ionization. We talked about being in the sun or submitting yourself to other types of radiation, things like that. And over time, your body can either repair and have normal cells, or it can go to apoptosis, or it can end up turning into a carcinogen, or it ends up mutating DNA. So there's a very fine line you have to decide what is going to help my patient versus what is going to hurt my patient. The idea behind the radiation treatment is that even though you have a bunch of mutated cells, and they are trying to grow over and over and over again, you hit them with more radiation so that the cell cycle becomes aware of the fact that its chromosomes are damaged. And what the hope is that you end up causing apoptosis for all those damaged cells. So the whole tumor should go away. Or you gave too much, you didn't hit that fine line, and you caused more cancer mutations. So the idea behind radiation treatment is try to have that cancer induce apoptosis throughout itself without causing more mutations and increase different cancer characteristics. Here's other ways of getting at the tumor. So these are using like Gamma knives. And you're getting the tumor from all different angles because it is hard just to get a tumor from just looking down into the body. So we got the differences between a Gamma knife using gamma rays or CyberKnife, which uses high X-rays, and then that bottom one, that C, is using protons. Point is those try to get as much angles onto that tumor without having to do something really invasive as going in and pulling out the tumor. So we know that when cancer is found, usually there are several forms of treatment. So having adjuvant therapy is just really saying, I'm going to have radiation and I'm going to have chemotherapy. Two types of treatments, hopefully working together to take care of that cancer. All right, some things you can do. So if you think about blood cancers, right now they're metastasized, right? Because your blood is all throughout your body. So it's not like it's sunk in a one set tumor that you can go in and remove. So it's you think, well, then how do you even treat something that's a blood cancer? The idea would be, we're going to pull out healthy cells from you, so bone cells that have the stem cells, and we're going to check to see that they're healthy, remove those, isolate them, and then cause irrigation, that's not the word, like radiation all throughout the body to try to kill the bad cancer cells, try to induce that apoptosis. And then when that's done, you put the good stem cells or the good blood cells that you had back into the body. So you take out some good stuff, have full treatment throughout the body, and then put that good stuff back in. So there are ways to spin down cells and to check that they're healthy and things like that. The other form you could do is that you get your blood cells from someone else. So the difference from autologous transplant, which is from yourself, or allogenic, which is from someone else. The bigger issue, though, is that if your body rejects it, your body has a really hard time wanting to accept anything foreign. It's trying to protect itself. And so if you were to do this, you would have to also suppress the immune system. When you suppress your immune system, you're more likely to get infections. If this is happening at a hospital, we know there's lots of infections that are at the hospital. So it's also hard to find a good match. Not everyone has a sibling that is ready to give you some bone marrow. And then it's also wicked expensive. Pulling it out, making sure that you can replicate those stem cells, why do I always want to say irrigation? Doing the radiation and then putting them back in. Very expensive process, but if you have some type of leukemia, that's the way it's going to go. Or that's one of the ways it's going to go. All right. So chemotherapy is using drugs to try to take away those cancer cells, but the really hard thing is chemotherapy goes throughout the entire body. So you're not only treating those cancer cells, but you're also going to end up affecting your normal cells. So here's that interplay of how much chemotherapy do you give the patient to where it's effective but doesn't kill them. So here are some different chemotherapies. These are the big popular ones. We got a whole bunch that end up causing adducts. So cisplatin, carboplatin, there's a couple more in the book. All they're doing is they're making that double helix form in a different way to where you can't activate DNA synthesis or gene expression. That should tell the cell to go through apoptosis. If you can't replicate correctly because of adducts, then stop the cell and head it through apoptosis. So fluorouracil is another one. That one blocks DNA synthesis. So if a cell is it's actually just stalled out. It's being held somewhere around S phase, stuck there forever with fluorouracil. What is fluorouracil's acronym? Hannah just laughed. So Hannah got it. Funny one. Anyhow. Okay. And then there's actinomycin that blocks transcription. You need transcription for making the proteins for DNA replication. DNA polymerase. To make more DNA polymerase, you need to have transcription active. So if you've shut that down by giving them actinomycin D, you can't make the things for gene replicate or DNA synthesis or any of the other big proteins needed for division. Okay, so lots of cool ways. There's also methotrexate. That's popular for breast cancers. Okay. Here is one. Here's a couple, vincristine and vinblastine, that basically make the mitotic spindle stop or not form correctly, so the cell cannot go through replication. So we can see in this figure, we've got two cells, or one cell that is about to divide, and you can see the tiny gray hairs of that mitotic spindle. I'd say they're probably in like anaphase. We're pulling those sets of chromosomes apart, and eventually cytokinesis will occur, and you'll have two separate cells. If we look down below, something that was treated with a taxane, which these are, we're seeing DNA replication is blocked. You're not going to go through the cell splitting like you should. So these cells should also then induce apoptosis. The goal is to tell the cell some things are very wrong, mitotic spindle isn't forming, our chromosomes aren't moving, so therefore go through apoptosis and kill off the tumor that way. So, okay. So this will lead to apoptosis. This just leads to cell cycle arrest, which will lead you to a silent death. So I talked about that fine line of how much are you treating the individual versus when does it become toxic for the individual. So all these chemotherapies that we know of, the first place that you start studying a chemotherapy intervention are in cells. So cells are the first place you go, and you start treating the cells with a certain chemical and see how many of those cells die with a certain concentration. Okay. So now you've kind of got your working concentration. If I give this amount, all of them die really fast. If I give it this amount, most of them die over a 2-week period. It's probably closer to that most of them dying over a 2-week period is the better choice versus all of them dying, because it's likely you'll then also kill all the cells nearby. Okay, so cells would be the first place to figure out kind of that dose response. How much do I need? And then you move into animals. So now we're treating a mouse. We're injecting them with the chemotherapy, and we're seeing how well does the tumor that they have. It's likely that we injected tumor cells on their back. They've got a tumor right on their back. Treat them with a chemical and see how long does it take for that tumor to go away, and or how long did it take for that mouse to die. If the mouse dies, we know it's too high of a treatment. If the tumor doesn't go away, then we know it's too low. So we have to find that sweet spot. And then we bring it on to the human, which we are not mice. Sure, we've got some characteristics of mice, but our body works different. So we have an idea that that drug might work in the human body, but its dose and its response are going to be different. So then there's all of those. This is that exploratory treatments that people get for cancer, and you're holding on to that. Maybe this one drug will do it. So they've got to take all the humans and really focus on what is the size of this individual, what age are they, what sex are they, as much info as you can get, because every human is individual, has individual characteristics compared to every other human. So for instance, I'm not particularly tall, but someone who is much taller than me is Caleb. So if Caleb and I were to have the same meds, we would not take the same amount, because he's got to take more than I do. And so even there, when we do these studies, we've got to figure out what is the best treatment for somebody who is, middle-aged, female, small, versus a healthy, young applicant, or what are you? You're an athlete, and you are. Go. Still, you got to go out there. So you can see how those factors would differ, and then what was the cause of the cancer? That might even have a play. So essentially, we're looking at two numbers. We have the effective dose, 50% that receive the dose at this level, the tumor will start to go away. But 50% of the people that receive this dose, right here, which is much higher, starts to where it's toxic to the body. I'm not saying you're dying at this point, but it's toxic to the body. The other number that we tend to look at is the LD. So LD is the lethal dose. So we don't like lethal doses. That's what we figure out in the animals. We figure out how much is going to kill that mouse. So the lethal dose versus effective dose. So that's what we're looking at in animals. And this chart is what we're looking at in humans. So the difference is the lethal dose we figure out in the animals, and the toxic dose is what we figure out in humans, to where you start getting really bad side effects. This shouldn't kill. You should not be killing the individual, but you should be seeing, okay, how much are they throwing up? How is their quality of life so interrupted? And they're just doing horrible with the chemotherapy. Then we know that that level is too high, and so we need to bring down that toxic dose. All right, so that's the human versus the animals versus the cells. And so things to think about when an individual is receiving chemotherapy. So yes, we know it's probably going to be a mix of drugs, and that you're going to have to do cycles of it, because those microtumors, maybe we're killing off you had surgery, boom, killed off the big tumor. Now those microtumors are giving the freedom to grow as much as they want, because they no longer have the endostatin, and the body is still producing growth hormones because you are still an alive human being. So you have to do cycles of treatments. But then it also says rest. And I think there's enough literature out there now that to say complete bed rest during chemotherapy is not the best course of action for the average adult. So there are plenty of people that I know now that receive chemotherapy and never take time off from their job. They don't take like a leave of absence. They keep working through it. And now they're saying you should actually continue to do physical activity because that's going to help the immune system deal with the therapies and the rest of the body, how it processes the poison of chemotherapy. All right, so here are the fun drug combinations. These are just some real popular ones that are out there. I'm not ever going to ask you to memorize this, but it's neat to see kind of like which drug combinations are good for which cancers. And obviously, you can see that there's lots of different drugs. Every single one of that's a drug, vinblastine we just talked about, right? So all of those come together. These four come together, and it treats Hodgkin's lymphoma really well. But think about how long it took us to figure out that drug treatment worked best for Hodgkin's. If we go over on this one, you can see prednisone, which is an active steroid, but it's really saying it's a version of cortisol. So our stress hormone in our body is often used as a chemotherapeutic drug for certain cancers. I don't know. We're not 100% sure on the roles of all of these drugs, so we don't know if cortisol is suppressing the immune system, which is somehow allowing one of these to work better, but we at least know that this drug setup is best for non-Hodgkin's lymphoma. So, Hannah, now you can kind of show different treatments that use cortisol. So we're doing an experiment on the HeLa cells and CHA cells to see if cortisol kills those cells off just as well. All right, so drug combinations. Here's just look at all the different drugs they have, but you may think coming up with those names are silly, but there's a reason. So all of the STEM names are going to mean, oh, it's this type of drug. So anything with an ANIB is an angiogenesis inhibitor. So you can go through that list. But so we talked about cisplatin. Well, it's named that way because it has platinum as making the adducts. Okay, so not super important that you memorize these either, but just note that the drug name does tell you about what it's doing to the cancer. All right, moving on to Gleevec. Gleevec is great. So the first thing is that, remember I said we don't know how all of these work, we just know that they work. Okay, so that's a mystery. When you're giving the body a drug and you don't really know how it's working, you don't know what else it's going to affect. So it's not ideal until you really know, we know this drug is going to get to these cells and have this action. Here we just know. We give the full body this, individual cancer seems to deplete, and hopefully the side effects aren't that bad. But Gleevec was great because it was very, very specific for a mutation. So this little green thing right here, that's Gleevec. This is also Gleevec in its chemical form. And what it does is it binds the tyrosine kinase. So we know kinases, remember how they could activate MYC genes or any other genes involved in gene expression or DNA replication. Okay, so your body has tons of tyrosine kinases doing their thing. There is a specific mutation that can happen to a tyrosine kinase. And so it turns into what's known as the cancerous form of ABL. The drug Gleevec, which is this little blue thing, it's this little green thing, the drug Gleevec will only work at that little, now I'm making it a red, that is where the mutation is occurring. So unless you have the mutation, Gleevec will not work. So now we're getting very specific. It's not going to work throughout the whole body because Not all your cells have that specific oncogene that is mutated. If you have the mutated oncogene, Gleevec is going to go in there and it's going to cause inhibition of the kinase, which then should lead to decreased DNA synthesis, decreased cell multiplication or division, and maybe through apoptosis. Okay, so that's great. Now we've got that happening, but a couple different things might happen. As your cells, as the tumor cells continue to divide, because you're not going to get all of them all the time. Gleevec's not going to work 100% of the time. There's no cancer drug out there that does. So you're going through cell divisions, and you could end up mutating ABL one more time. So if ABL has more mutations, then it can't bind Gleevec anymore. So more mutations, less binding affinity. For Gleevec. And I spelled everything right, even though it's messy. Okay, so that kind of sucks. So it only works in a certain stage before other mutations have occurred. And then, this is so amazing. Cancer cells figure out how to get cancer chemotherapy drugs out of them. So you remember, you got the cell membrane, so it's a big sack of stuff, and the chemotherapeutic drug enters through the plasma membrane, so it has to have entry, and then it should be doing its job inside. But the cancer cells have figured out how to take that cancer treatment and push it back out. So it's got these drug transporters that are going to push them back out, making it that the Gleevec isn't going to have its chance to bind to that mutated ABL gene or ABL protein. So that's not cool. And many other cancer cells can do this with other drugs, so that's why it's called multi-drug resistant. Several drugs go in, and then they just get kicked back out. So Gleevec is great because it's pinpoints that cancer, that mutation. Shouldn't hit the rest of your body. But then the mutation may acquire more mutations so it doesn't work, or that cell figures out how to kick the drug out through those multi-drug transporters. All right, and then kind of ending where we began, looking back at, we had that stain for the breast cancer cell. I'm going to go back here. And we were staining here if it had estrogen receptor. So if it has estrogen receptor, this is why we care if it's ER positive or negative. All right, so estrogen, that's a steroid hormone, and it is anabolic. All right, so how do you determine if something is anabolic versus catabolic? It's just anabolic. When you get that treatment, you start to grow. Anabolic means putting things together and building it. Catabolic is breaking things down. So when you receive estrogen, it causes cell division. Another example is testosterone. That is also anabolic. Okay, so you can see we've got two hormones that are anabolic. Cortisol is not anabolic, just in case you wanted, well, it's not very anabolic. So your sex hormones are anabolic. Cortisol and aldosterone are not considered anabolic. All right, so the point is here that if the breast cancer that is being tested is ER positive, which should mean grows with estrogen. So if you have an ER positive breast tissue, then we know if we give it estrogen, it should grow. So if we can somehow block estrogen from going into those cells, or block the receptor or something like that, the estrogen receptor, then maybe that's a way to treat it. So here's this drug called tamoxifen, and that essentially blocks where estrogen would bind. So estrogen can no longer work, so tamoxifen blocks estrogen, and so cell division should not happen. So that's great, and that's why we try to figure out if you're ER positive. If you're ER negative, you could give them tamoxifen, but it's not going to do anything because the receptor's not there. You have to have that receptor to treat with tamoxifen. So flutamide was another one, and that's a testosterone anabolic inhibitor. So again, you don't, in the testes, if there's high levels of testosterone for that cancer, then it's going to keep growing. So if you create something that can block testosterone binding, great. There's also, this one is another testosterone one, and that's what's called chemical castration because it blocks the ability of all testosterone throughout the body. So this is a way, I think, that they've treated pedophiles in jail because it totally kills all sex drive. All right, next one, Herceptin, and this one we kind of talked about, but this is great. And if you read Breath Becomes Air, this was the treatment that they were trying to use on this individual. So we know it's good. It can treat breast cancer. I still cannot spell that. Breast cancer, and also small percentage of lung cancer. So in Breath Becomes Air, this was a doctor who realized he had cancer, and he was looking for different types of treatments, so he knew he needed to test his tissue to see if there was epidermal growth factor receptor, and he found out, I believe he found out he was, and then so they used Herceptin, which, like the other inhibitor, it basically binds it and no intracellular activity. So it doesn't even get inside the cell. It shuts down the receptor on the outside, and there's no intracellular activity, so we're not going to go through cell division. So another great drug, very specific, so it should not hit all the rest of your cells. I think, again, in Breath Becomes Air, that after a while it no longer works. So it worked for a little bit, and then it stopped working. All right, and so last thing to note, I think it's second to last thing to note, one way to go about chemotherapy is just to block angiogenesis. So if the tumor cell can't get blood to its area, so it's not going to get nutrients and it's not going to take away lots of the waste materials. So having a drug that blocks angiogenesis is a great way to treat that, especially after you remove the bigger one. However, I'll start with Lakin. What would be some bad thing about blocking angiogenesis?
I don't know, can they block angiogenesis in only the tumor?
Nope. So, not cell specific. Okay, Christina, you get another one.
Does it make it metastasize quicker?
Well, if we no longer have blood vessels, it wouldn't metastasize as fast. Yeah, so it's good for stops or should slow metastasis. Tyler's gone, so Jillian, it's over to you. What else would be bad about giving a human angiogenic blockers? Yep. Repair internal and external injuries. You'd be surprised how many internal injuries you have, especially when you're older, with high blood pressure that can cause blood vessels to rupture all the time. Obviously, we know that myocardial infarctions, heart attacks, you need angiogenesis to repair that so that the collateral routes go around where the dead area was. So if somebody had really bad atherosclerosis and had cancer, giving them a blocking of angiogenesis is probably the worst. I don't know if it's the worst thing, but it's not helpful for sure. So yeah, there can be good and bad for that. And then let's go to this one. Alternative medicine. First off, does anyone know some alternative medicines for cancer or for anything? What do you got? I'm going to ask, who am I asking? Paige. Or when you think alternative medicine, does anything pop out?
Like tea.
Tea. Yeah. Because green tea is supposed to be an antioxidant. And we know that free radicals lead to reactive oxygen species, which leads to inflammation causing tumor issues, right? DNA issues. So if we have an antioxidant fighting those free radicals, then that would help. So that's one. I heard shrooms. I don't know. I can't. How do you spell shrooms? Even on a good day, I couldn't spell that word. How would?
I think it's just mushrooms without the MU.
You assume I can spell mushrooms. I don't know why you would think that. You wait. When you have to teach a class, all spell check goes out your head. You have no idea how to spell anything.
I think it should be spelled the first way. I say we make a petition.
Yeah, let's make.
No,
Let's- We'll contact SGA, see if we can get that going.
I agree. Well, we can talk to these two are part of SGA, so.
There you go. Help. We need to change.
Shrooms that make you feel good.
What these are?
Yeah.
Okay. So there you go. There's a alternative medicine. We do know that they've been used to treat cancer. What are the side effects? Can't drive a vehicle while on shrooms. Right? Can't or shouldn't. Right? So we know it could affect your mental capacity. So there's like tons of alternative medicine out there. There's lots more when you consider Eastern medicine versus Western medicine. America tends to tightly control their drugs. So the FDA tightly regulates. Other countries are more relaxed, and so there's more exploratory studies on alternative medicine in other countries. That's good or bad, because we need more treatments, different treatments, so we should be exploring these. But then bad because widely treating people with alternative medicines, not knowing how it's going to react on the body, can be bad. Hannah, do you got anything else about alternative medicines?
No, I was just kind of thinking like manipulation, even.
Like how?
Like, not like chiropractor stuff, but like.
Yeah. So, that's an A. I can't help that you can't see that's an A.
That's an S.
What is that? Girl.
See how long it would take for me to teach if I had to really spell out words?
You wrote that really nice, though.
Thanks. Highlight of my day. Anyway, so yeah, there's manipulation. So some think that acupuncture, things like that can take care of the cancer. I'm not, I don't know that there's definitive articles out there that say yes or no, but I don't know that sticking a needle in you is going to take away your cancer. But some people say, no, that's the way I'm going to be healed. Other things would be if it reacts with drugs. So if your chemotherapy does XYZ and creates some type of product that interacts with the alternative medicine, and the doctor's like, what is happening? This should be working, and here you are super sick or something, and then you tell the doctor, oh, but I'm taking this antioxidant or something, and so then you figure out, oh, it interacts with your drugs that are supposed to be helping you. Yes?
Would you say like certain diets?
Sure. There are lots of diets that people start. They do juice cleanses when they hear they have cancer. There's lots of different things. So even just what goes through our daily life when we have to have certain treatments, we need to tell our doctors everything we're taking, literally everything, so that they see if there's any counter actions. All right, that's it. Any questions? No questions.
Wait, are we like done?
You're done. You're done till the 21st. This is what happens when I don't do quizzes in class and I don't ask questions. So I'll ask more questions next time. So Tyler even missed half of it.
I said we do it again.
That's awesome.